Arabidopsis is a widely studied model plant, encompassing genome, organelle, cell, tissue, and subcellular proteome studies, as well as plant developmental processes and responses to biotic and abiotic stress using various quantitative methods. Research using Arabidopsis as a model plant has also been conducted to observe plant growth direction. In Arabidopsis thaliana, auxin transporter proteins regulate phyllotaxis, causing auxin accumulation to occur only at a certain minimum distance from established primordia, thus determining the position of new primordia. The leaf primordium pattern in Arabidopsis thaliana demonstrates that the amount of auxin-based protein pinformed can influence plant organ formation regardless of its form or developmental origin. Auxin levels and growth are correlated because auxin-based protein pinformed, an auxin transporter, influences the direction of microtubule arrangement in cells. Research has shown that meristem and flower primordium patterns are also influenced by auxin biosynthesis. In Arabidopsis, without auxin biosynthesis and transport, shoot meristem growth is disrupted. Polar auxin transport regulates the direction of leaf growth and leaf vein patterns by forming auxin convergence points. This triggers the formation of leaves and main leaf veins. Auxin is a plant hormone that plays a crucial role in the early phase of organogenesis in plants, particularly in the shoot by maintaining the balance between stem cells in the central shoot apical meristem. This pattern is primarily regulated by polar auxin transport, also known as directional auxin transport. This mechanism is controlled by the auxin efflux protein pinformed, suggesting that the auxin gradient generated by the pinformed protein is a general and sufficient mechanism to trigger organ initiation. In Arabidopsis, auxin acts as a morphogen, utilizing polar auxin transport to independently form patterns. When polar auxin transport is inhibited, the vascular pattern changes during phyllotaxis. This explains that the direction of auxin flow is a factor that can determine the direction of plant growth. The direction of auxin flow is determined by the orientation of pinformed proteins on the cell surface or subcellular polarity. The TMO5 genetic pathway or auxin-activated gene regulates the polarity convergence of auxin transport proteins toward organ formation. These results align with the assumption that proper auxin accumulation depends on the direction of auxin transport. The role of auxin transport in determining growth direction must be integrated with auxin biosynthesis mechanisms. Auxin is produced locally in the shoot apical meristem and contributes to pattern formation and differentiation. The direction of auxin transport is also influenced by the mechanical structure of the cell. The orientation of auxin transport proteins is closely related to the orientation of cortical microtubules. These results indicate that mechanical stress and changes in cell shape, associated with the cell wall, can regulate the direction of pinformed, and vice versa. Auxin can trigger the phosphorylation of myosin, a cell motor protein, which plays a role in influencing the movement and position of pinformed. Genetic mechanisms contribute to regulating growth direction, but this pattern is then refined by physical mechanisms such as microtubule orientation. This mechanism explains that phyllotaxis is the result of an interaction between gene regulation and cell mechanics. primordium formation is determined not only by growth direction but also by timing. The timing of leaf formation is controlled by a mechanism that works between auxin and the signal peptide EPFL2, a peptide that regulates growth direction. Auxin plays a key role not only as a growth regulator in Arabidopsis but also as a morphogen, or pattern-determining agent that shapes plant structure. This is because auxin distribution is controlled by polar auxin transport. The polarity pattern of pinformed determines the direction of new leaf primordium formation. When the direction of auxin transport is disrupted, the phyllotaxis pattern can change. The primary role of auxin in Arabidopsis is to determine the pattern of organ formation, not simply to promote cell growth.